CEO. MaxCyte is a non-viral cell engineering company working with cell and gene therapy developers, engineer cells all the way from concept to clinic and now through commercialization. This is a forward-looking statement that the attorneys make me put in there, so bear with it, but now you're ready, we'll move on. So what is MaxCyte? MaxCyte, we're now celebrating 25 years, right? We've been doing this for 25 years where we have become truly the pioneer for non-viral gene delivery. We have a leading electroporation platform from concept through clinic through commercialization, where we, through our flow electroporation, transfection technology, we're able to work with clinical developers all the way from early concept through the clinic, through commercialization, and we're happy and proud to say that last year, the first non-viral cell therapy was approved utilizing our system to engineer the cells. That was Vertex CRISPR's XSL Casgevy approval. Electroporation can be used in a wide variety of applications. So in addition to cell and gene therapies, you can have viral vector production, cell-based assays, antibody production, even vaccine development. But in this presentation, I'm really going to focus on what we do in the cell and gene therapy space, what our technology is, what our scientific expertise is around that technology, and how we work and partner with companies in the clinical development space for cell and gene therapies. So cell and gene therapy development, even before, obviously in the last 10, 15 years, has really come about, something that was never anticipated when the company was first founded. The cell therapies have now become, the modality of treatment for many diseases, whether it's genetic diseases, rare diseases, oncology, solid tumors, hematological cancers, even immune diseases that we're now autoimmune diseases that we're now seeing take hold with cell therapies. And there are two ways you can engineer cells. It's in vivo or ex vivo. And in the ex vivo space, roughly 40% of ex vivo delivery is done through non-viral genetic editing. And truly, the only way you can do non-viral genetic editing is electroporation. And we have that platform that's been pioneered for the last 25 years, specific for cell therapy non-viral delivery. Electroporation uses electropulses, where you're electroporating the cells, and you're temporarily and reversibly opening up the cell membrane to allow genetic payload to enter into the cell. That's done in a very efficient manner. It's done in a very soft and gentle manner where cell viability on our platform is higher than other platforms. And we're seeing more of a trend towards that non-viral cell engineering with new gene editing platforms, with allogeneic donor cell engineering that requires greater scale, with increasing complexity, where now you have many more edits and different cells being edited, whether it's T cells, B cells, NK cells, TCRs, TILs. That's lending itself to non-viral cell engineering, which is what our platform has been designed for. So where do we play in the cell therapy ex vivo workflow? We have a closed cGMP cell therapy engineering system that allows us to play agnostic to upstream and downstream technologies to the MaxCyte EP system. So whether, regardless of whatever CCV system you're using, regardless of whatever expansion system you're using, we have the only EP developed specifically for cell therapy, for clinical development and now commercialization, as I mentioned, with the Casgevy approval last year, that was engineered on our cells. We have proprietary EP parameters that our scientists have built over the last 25 years, where we have over greater than 100 protocols optimized for various cell types, various genetic editing payloads. We have seamless scalability, where we can take developers from very early discovery all the way through commercialization. And as I mentioned, we've built a scientific team, both internally, with our application scientists, process development scientists, as well as field application scientists, globally. That's in the US, in Europe, and in Asia, where we work with companies as they optimize their processes. How do we do that? So we do it by really having the only systems that can scale from very early research all the way through commercialization. You'll see here our Expert ATX that allows companies to start working on the concept and discovery of their cell engineering, where you can transfect as little as 75,000 cells all the way through that seamless process scalability, where you can use our Expert flow electroporation, where you can transfect up to 20 billion cells. And that's all done on the same parameters, the same protocols. We're the only company that have that ability to scale a company and really accelerate their clinical development timeline. But it's more than just accelerate. We're also giving them viability of a commercial product even early in the research and discovery phase. So whether you're using our systems in early research, that same protocol, the same processes, you're going to be able to utilize all the way in the clinic and in commercialization. So in essence, we provide therapeutic developers the ability to have a commercial product or a commercial process, even at the research phase. Part of that is obviously with the scalability that we have, and we've built our processing assemblies to allow companies to have various levels of transfection, from, as I mentioned, from 75,000 cells to 20 billion cells, even with our VLX, which can transfect up to 200 billion cells. And it's all done with processing assemblies that can be bio-welded to the CCB systems pre-EP, as well as to the bioreactors post-EP. So you can have a truly closed system through our range of processing assemblies. So what does that mean for developers, and what does that mean for clinical companies pursuing autologous cell therapies? It means you have a clinically validated and really now commercially validated. With the approval of CASGEVY, we now have the only commercially validated electroporation system that's very easy to use. It sits nicely in a clean room, has reproducibility from clean room to clean room. And we have over 1,000 patients that have now been dosed with cells engineered on our system. It also means that reproducibility, where from donor to donor, you have the same editing efficiency, whether it's on knock-in or on knockout efficiency, as well as regardless of cell type. You have the same reproducibility, whether you're transfecting T cells or TCRs or TILs or NK cells or B cells. And we also have focus on ensuring that these cells are precious, right? So we have minimized cell loss. We have volume loss of less than 10%, and many of our customers are saying that they're seeing less than 5% cell loss when processing cells through our system. We also support a decentralized manufacturing process, where we have tech transfer from CDMO to CDMO, whether you're transferring from Europe to the US or vice versa. We are able to support that, that process and train CDMOs on our system, on the process, to ensure that we have the same reproducibility of processes across the globe, from patient to patient, site to site. What does it mean for allogeneic therapies as well? So obviously it's twofold. One is with the ability to process and transfect up to 20 billion cells on our system, you have the ability to really reduce COGS, where from one run, you can create almost 4 times as many batches per run as you would on any other electroporation system. What does that mean also? It's not just COGS, right? It also means you can have better quality control, because rather than multiple runs to create batches, you're really running everything on one run, minimizing the chance of quality contamination, and you're not pooling in any way either. So on one run, you can produce the majority of the manufacturing batches you need for an allogeneic therapy. This is a slide if I was going to talk about, even though I talked about our EP system, electroporation system, that means the most to us and why we partner with cell therapy companies. In addition to our instrumentation, which is fully closed and scalable and 21 CFR Part 11, that's the basics of what we do. Really what we do is everything else. It's that scientific and technical support. It's those 35 plus field application scientists, application support in the labs, process development support in the labs, specialized around the cells, specialized around the gene editing materials being delivered into the cells. We do that with one mindset and one mindset only, which is an obsession with ensuring our customer success. That really is the culture of MaxCyte. And in addition to that, we have regulatory support for cell therapy developers, whether it's early on discovery all the way through the clinic and commercialization. We have a master file that our cell therapy developers can reference and has been referenced in the first and only non-viral cell therapy approved with CASGEVY. That same master file is in the US, is in Japan, is in EMA, through master files or technical files, and we provide on a country-by-country basis regulatory support as cell therapy developers are developing their products. With that, we also have built a top quality system in our Rockville facility in Maryland, where we have brought in all our manufacturing in-house. All of our instruments, all of our processing assemblies are manufactured in-house. In addition, we have a global partnership support where every partner of ours has a touch point where they can reach out to one person, and whether it's a quality issue, a regulatory issue, a scientific issue that they're having, where for whatever reason, if they're having CDMO issues, or if they're having engineering issues, we're there for them 24/7 to ensure that we can support them in any way. Again, it's that obsession with the customer success that's really MaxCyte's bread and butter. That's our DNA. What does that mean for MaxCyte? What does it mean for cell therapy companies? We're very proud of this slide for 2 reasons. One for us, but one what it means for the industry and for our partners. We have now 27 signed clinical and commercial licenses with cell and gene therapy developers. This has resulted in 16 current programs currently in the clinic utilizing our system to engineer the cells in a non-viral, efficient, and viable manner. That's resulted in one commercial program with Casgevy by Vertex and CRISPR. You can see here from 2017 was our first one with CRISPR Therapeutics to the last one we just signed right now with B Biopharma for the editing of B cells. These therapies are across modalities, whether it's allogeneic or autologous, as I mentioned. It's across clinical development timeline as well. We have phase 1, phase 1/2, even pivotal, and commercial program now. And what you see here, you probably notice there's more than 16 dots compared to the other slide. We also have investigator-initiated trials utilizing our system to treat patients in a clinical setting. So we're able to show that concept to go from really early research to clinical all the way through commercialization using a scalable, optimized process. These are some of the genetic diseases that we enable on our system, whether it's sickle cell and beta thalassemia, which was approved late last year, as well as solid tumors, infectious diseases that are being pursued with cell therapies right now, hematological malignancies, as well as autoimmune diseases that we're seeing truly take hold. Regardless of gene editing tools, our system is designed to ensure that you can work with base editors, Cas, or any other gene editing tools. I want to take a few minutes just to talk about one kind of special company that we've been working with since 2019. It's KSQ Therapeutics. So KSQ Therapeutics is engineering tumor-infiltrating lymphocytes, or TILs for shorts, and for solid tumors. TILs have the advantage over traditional CAR-Ts in the sense of they already have shown tumor specificity. These are immune cells that have already made it to the solid tumors of patients. However, TILs are also a little bit harder to engineer. KSQ has a proprietary and a simple process where they're able to reduce the tumor-to-vein production timeline, and they needed to utilize our system to ensure that the transfection process has the ability to have the efficiency and the reproducibility that other transfection processes did not have. We began working with them in 2019. It's not showing everything there. I apologize, but I'll walk you through it. They had a few challenges. So in 2019, they were using a different transfection system to try to engineer their TILs. They were not getting the efficiency of the editing they were requiring. They were not getting reproducibility and the consistency from donor to donor, as these are autologous therapies. In 2019, using our small-scale ATX, they were able to have higher knock-in efficiency of the edit, as well as the ability to have reproducibility. In addition, we have that ability, as I mentioned, to work with companies to have a process that's really a commercial process even before you're ready for the clinic. In November of 2019, we worked with them, introduced our flow electroporation system, the Expert GTX, where we were able to dose, whether it was 400 microliters to 1 milliliters of cells from donor to donor with the same efficiency of knock-in, with the same reproducibility. In 2020, after we signed that SPL with them, they actually had an issue. This was during COVID, where they had CDMO shutdowns. As we know, COVID was a tough time for our industry. And what we're able to do through the support of the entire organization is we're able to work transferring our system, transferring the process, training a new CDMO, first from Europe, then into the US. We had to do that again, and we did this all seamlessly with no interruption to their processes. It's part of what we do. It's the support that we do. It's that what I talked about, our ability to ensure that we have that obsession with customer success no matter what. And regardless of whether a company is in early clinical, late pivotal, or commercialization, we show the same support throughout. And in November of last year, they were able to successfully file an IND, and they're currently in a phase 1 program right now for KSQ 001 for the treatment of solid tumors. This last slide, if it lets me bring it up. This is some of what our partners say about us. And without going into each and every quote, it talks about the fact that we have a commercial product. It talks about the fact that we have a transfection process that can get you from research all the way to the clinic. You, in essence, have locked in your process working with us early on all the way through commercialization. And we're very proud of this. And we're proud of what we're doing with the industry. And we're proud of what the industry is doing. And we want to make sure we can do everything we can to speed up their process and give them a really viable product at the end. This is a partnering program, obviously partnering conference. So this next slide, Shalmer Nargez, who's our head of business development, is here at the conference. Feel free to reach out to him. Feel free to reach out to me as well if you need anything. Thank you.